Welcome to the Adelaide Biomed City Minireview webinar series. My name's Andrew Zannettino and I've got the good fortune of being your chair this afternoon. Before I introduce our first speaker, I'd like to acknowledge the Kaurna people, the original custodians of the land in which we meet here today. I'd like to pay my respects to elders past, present and emerging. Really important part of this is engagement with our people online. And I'd like to remind people that you can use the Q&A function on the Zoom window just at the bottom. And that'll allow me to relate your questions to our speakers. And again, I really encourage you to do that. This afternoon, we're actually very fortunate to have two speakers, Dr. Mark Corbett and Dr. James, or Jimmy Breen to his friends. Our first speaker is Dr. Mark Corbett, and just by way of a bit of background, Mark is based at the University of Adelaide and in the medical school. He's a specialist in DNA and RNA sequencing techniques, which he has used to implicate over 30 new genes in neurodevelopmental disorders. So collectively, Mark's discoveries have led to genetic diagnoses of thousands of individuals living with neurodevelopmental disorders around the world. So this afternoon, his presentation is entitled, "What does it mean to discover a gene?" So I'd like to welcome Mark. So thank you, Mark.
Yes, thanks, Andrew. Today I'm here representing the Neurogenetics Research Group and also the Australian Collaborative CP Research Group. And we're both based at the University of Adelaide Medical School. And really I'm here standing in primarily for Joseph, who was kind enough to give me this opportunity. If anyone has any questions, feel free to contact me after the presentation. So what I really wanted to talk about today is we're primarily involved in gene discovery. And if you go through the internet and do some Googling like I did over the weekend, you can find an amusing number of genes which scientists have discovered, all to kids' uncombable hair through to genes which apparently make you skinny. But what is really being said when you see these kinds of headlines is that people are discovering genetic variants. Everybody's got the same set of genes, but not everyone's got exactly the same sequence within those genes. And those genetic variants directly influence everyone's health, but the effects of those variants can be either large or small. So small might be how combable your child's hair is, but large could be whether you're affected by a severe neurodevelopmental disorder. And that's our focus in our laboratory. So neurodevelopmental disorders are characterised by developmental deficits that produce impairments of personal, social, academic or occupational functioning. So some examples of these are intellectual disability, epilepsy, cerebral palsy or autism spectrum disorder. And while I'm on this page, I'd really love to draw your attention to this website, Positive Exposure, which is an initiative of a photographer to really bring to light the beauty of the people who are behind and the families that are behind these disorders and bring them out of the darkness and into the light. So please have a look at it. It's a great initiative. In terms of our laboratory and our impact on the field of neurogenetics, we've discovered collectively through collaborations and also within the lab over 250 genes which we've implicated in neurodevelopmental disorders. I'm going to use this abbreviation NDD for that. That's involving over 5,000 South Australians in our research programs, and that means that over 500 families have received a new genetic diagnosis, and that's just directly from our research. Once a gene is found, it can't be unfound. So these lead on to genetic diagnosis further down the track. There's just an example of some next generation sequencing, which is primarily the technique that we use, and we segregate these variants through families in order to establish their pathogenicity. So I just want to share with you this email which we received, and really to say, what does it mean for families when we discover these genes? And just to say that this mother of this child, who was affected by a variant in the KCNA2 gene, which I just showed you a variant of on the previous slide, and that results in epilepsy and also a tremor-like disorder, but that you note that she's still pleased and enjoys spending time with her son, and that also the research that we're doing here and similar research being done around the world helps to link families together through these genetic discoveries. It gives them an answer for why their child is affected, and the flow-on effect helps them direct their efforts towards the next stage in trying to find the next solution for their child, and takes away that guessing. Not only is there this translational effect, but also we're learning every time we discover a new gene involved in a neurodevelopmental disorder, we're discovering new molecular pathways linked perhaps to cognitive mechanisms. So here are just some examples of some capabilities within the lab. So through Dr. Lachlan Jolly, who's a fantastic neurodevelopmental and cell biologist, and you can see some of his fantastic immunohistochemistry here, studying in this example an mRNA nonsense-mediated decay pathway, which involves these 3 genes, which we've found variants of in all 3 in children with intellectual disability. And then also the protein complex, T-Rex complex, which allows RNA to be exported from the nucleus. And this is work led by Dr. Raman Kumar, who's an excellent molecular biologist. And we're actually translating a deletion, mutation, which we found in this gene, now to a new mouse model, which you can see this little tiny fella on this side actually has that mutation while his brother does not. So I want to spend the last couple of minutes on this very short talk talking about some of the translational impact of our work. So we work with Alistair McLennan, as I said, on cerebral palsies. And at the moment, genetic testing is not routinely offered for children living with cerebral palsy, even though you would call this a neurodevelopmental disorder. So Alistair and his team just established the Australian Cerebral Palsy Biobank, has over 500 families who are living with CP enrolled in that, and we have DNA from all of them. We have about over 500 patient cell lines, mostly derived from patient blood. We've done most of the sequencing on most of the individuals in that bank in some form, either exomes or genomes. We've done quite a bit of RNA sequencing and also some methylation sequencing. So when we did the exome sequencing studies, and we've replicated this result in 2 independent exome sequencing studies, approximately 14% of these individuals have single nucleotide variants that can explain their disorder. And a further 3.77% have pathogenic copy number variants. When Claire Van Eyck in our lab, who's also a bioinformatician, molecular biologist, performed an RNA sequencing study, she added an additional 5% of variants. So up to 1 in 4 of these individuals actually have a genetic variant which can explain their disease. These results are being used now to change clinical practice in the diagnosis of cerebral palsy. Claire also found through her RNA sequencing study, there's an overlap between the transcriptomic changes in the cell lines of these individuals in cerebral palsy, and it's quite related to autism spectrum disorder. It's an interesting observation. So now I just want to quickly show you an example of how we can take, and using the CP Biobank data, how we can also extend our ability to find a genetic diagnosis by overlapping multiple omic data, if you like. So here's some information. Here's RNA sequencing data. And here we've scaled the counts for this particular gene, DIP2B, and we've just normalized them all. So you can see this box plot indicates a normal distribution, the box and whiskers. And then occasionally we have outliers, as is indicated by the blue dot. And that shows that this patient's gene expression is incredibly low compared to the normal population. This little pink arrow here, just to highlight that. When we looked in the DIP2B gene though, however, we did not find a pathogenic variant in our exome data. So we had the methylation data for this individual, which is indicated just at the bottom of this collection. So these are reads which were sequenced and converted to detect methylation. When it's blue, there's no methylation, and when it's red, there is. So this is overlapping just the promoter region of the DIP2B gene where we find this change in gene expression. And we can see in the patient only, there's a half of the reads are red while half of them are blue. This indicates that one of the chromosomes for this individual has gene expression being shut down, most likely due to hypermethylation. But we still couldn't see on the exome sequencing data the mutation here. So now we've set up a nanopore sequencing. So this is a long read DNA sequencing facility. I'm happy to talk more about that if you have questions, which allows us to read much longer lengths of DNA sequencing. And we targeted the DIP2B region, and we were able to identify a 2.5 kilobase insertion of a 3 base pair repeat. This is a known repeat expansion, and when we correlated those reads, so some of them have this browny color, which indicates the repeat. When we correlated those with the methylation data, also extracted from the long reads, we see that they're the reads which are causing this red color and are being hypermethylated. So this allows us to understand what's causing this downregulation. This is a known pathogenic mutation, and that's resolved this patient. So here's our group, lovely bunch of people. We do have a few errant cancer biologists in here, such as Wayne Tilley, and so this is our level 8 Adelaide Health and Medical Sciences building Christmas party, but these are the people behind the work I've presented you today. Thank you.
Thanks, Mark, for a fascinating presentation. While we wait for questions to come through via the Q&A function of Zoom, I was just really curious about the pathways that you've been identifying. Clearly, there would be a desire to see whether these would actually be actionable using sort of a therapy-based approach. Can you give us a sense of the number of pathways that you've identified, whether there are existing or they are indeed actionable targets by sort of medicinal chemistry approaches?
Yeah, that's a fantastic question, Andrew, and it's certainly something that we've got a lot of focus on. So in terms of pathways leading to neurodevelopmental disorders, pretty much any pathway you can think of in a cell is in some way involved, mainly because they all at an upper level result in a change in metabolism in the brain, and it's very susceptible to changes across the board. So you'll see the RNA metabolism is very interesting, but also transcription factors, intracellular signaling molecules, cell-cell adhesion, all those sorts of things. There are several places you can go with these particular genes, and then you can look to see whether there are any drugs or clinical trials associated with any of them. One of the, so the NIH has a collection of, or a database which links genes to pharmaceuticals. There's also a website called treatableid.org, which again is specifically targeting intellectual disability and linking different groups together. So there's lots of different ways in which you can treat these disorders. Some of these neurodevelopmental disorders are metabolic, so they're perhaps more easily treated. Some of the more genetic ones, we're going to need to use some fancier methods. But yeah, that's definitely an area of interest for us.
Thanks, Mark. If I may, while there's no questions coming through, I might ask a second question. You mentioned you've developed 500 or cell lines from 500 families. Can you tell us a bit more about what you mean by developing cell lines?
Okay, so these are lymphoblastoid cell lines. So they're immortalized, Epstein-Barr virus immortalized white blood cells, or B cells, and they're just taken from patient blood. You can take those and then also convert them through to, say, an iPS cell line, but that's a little bit more involved and a lot more expensive. So we were able to get most of those done through a facility called the Genetic Repositories Australia, so GRA. But unfortunately, that facility is no longer funded. So we have that biobank stored now in Adelaide University. It's definitely the largest one we know of internationally for cerebral palsy.
Mark, I have a question coming from Paul Thomas, who has asked, is nanopore sequencing useful for identification of point mutations in long reads? Seems to be quite a lot of errors in our experience.
He's correct. For a moment there, I thought he was going to tell me off for not acknowledging him making the THOG2 mouse. So you can do it. So we've actually done it. We get about 85% recall on single nucleotide variants. It really depends on the sequence context. If you're looking at insertions, deletions, your recall is much, much worse. So that's like the ability to rediscover a variant that you already know. It goes down to about 60%. It can be done. And if anyone's keen, we're keen to collaborate. The nanopore stuff is my most exciting thing at the moment.
Thanks so much, Mark, for a fantastic presentation. We'll move on to our next speaker, Dr. Jimmy Breen. Most of you who are watching this, I'm sure, know Jimmy. Jimmy is like oxygen, he's everywhere. Jimmy is a computational biologist that leads bioinformatics at South Australian Genomics Centre, located at Zannettino. Jimmy has had an incredibly diverse career in genomics and bioinformatics research in both academic and not-for-profit institutions in Australia and Europe. Jimmy is also an advocate for bioinformaticians across Australia and was elected the president of the Australian Bioinformatics and Computational Biology Society in 2019. It gives me great pleasure to introduce Jimmy, who will today tell you about partnering genomic and bioinformatics at the South Australian Genomics Centre, or SAGC. Welcome, Jimmy.
Awesome. Thank you, Andrew. This particular presentation, I'm really going to do a little bit of a sell in terms of why you need to be partnering genomics and bioinformatics together, and sort of really a demonstration of capabilities that we have at the SAGC that started last year. Let me just do that. So yeah, so I'm just going to talk about bioinformatics and really what we describe as sort of an analysis bottleneck that a lot of people need to overcome in a lot of their projects. Partnering genomics and bioinformatics, which really does overcome a lot of those particular issues. A little bit about the South Australian Genomics Centre, although I know that this has been highlighted in a few webinars before. And really the services and sort of the expertise of my team and partnering with people within the Adelaide Biomed City, but not just the precinct, but also across South Australia and Australia and internationally. So I'm sure many of you have seen this particular slide before, but really the context behind it is quite important in the fact that genomic data is actually relatively cheap to produce. If you're an old fogey like myself, where we used to do create genomes from back-by-back sequencing of Sanger, we're looking at an ordering of like $100 million per human genome, and that's kind of steadily decreased until about 2009, 2010, where we had this introduction of next generation sequencing machines, which really has helped a lot of things, including the things that Mark talked about previously. So this massive development drove a bit of a problem in the fact that every person can now get their genome sequenced for around about $1,000, but really it's the expertise in being able to analyze that that's really the bottleneck that delays bioinformatic sort of results and publications, and as a consequence, money and clinical sort of outcomes. So why do we have this bottleneck? So really, analysis is actually quite expensive. It requires personnel with specialized multidisciplinary training. And really, if anyone listening right now has had to recruit a bioinformatician, you know it's very, very hard because there's a large diverse, bioinformatics means a whole lot of different things, and people have skills in sort of lots of different parts. And it may not, be perfect for the particular role you're looking for. Also, I've been a massive advocate for that. Bioinformaticians are not particularly well trained within our academic system. And so really, academic or non-academic, the training available for people with the right skills to do genomics, high throughput sequencing data analysis is actually fairly limited. And really one of the problems is that these are all the things that encompass bioinformatics. You've got a little bit of statistics, you've got a little bit of computational science, and then you've got a lot of biology. Now, really, the intersection of all those is really a bioinformatician. And really, there are aspects or other areas such as biostatistics, computational biology, and data scientists that kind of are on the periphery. But really, we're looking at a person who could not only interpret data, look at the statistics behind it and apply it, but also and a little bit of the interpretation of the data which comes through, because that really makes the project move forward a lot quicker. Not only that, another problem is that genomics and bioinformatics requires, especially with high throughput sequencing data, requires incredibly expensive computational resources, such as a server farm or a high throughput, high performance computing facility. Now, a lot of this has now been put into the cloud, and which has driven somewhat of the costs down. But another problem with that is that because sequencing machines produce an incredible amount of data, sometimes on the order of 2 to 4 terabytes per sequencing run, it means that if we're pushing data up into the cloud and bringing it back down, there's a cost involved with that. And it also reduces the performance of the programs that we design to do all these particular things. But perhaps the most important is it's the least funded part of a sequencing project. So personnel focus on producing samples, quite rightly, because that's really what you get the funding to do, to look at a particular biological question. But really, a lot of the time, there's nothing left for the analysis. Often this is left to PhD students with no computational support or mentorship from a person such as myself, because there's not many of them within an academic or institutional system, because they're quite rare. But also it delays research and clinical outcomes. And that's really the most important thing I want to focus here today. So really the best way of overcoming this is partnering genomics and bioinformatics and as a service, not necessarily within a particular research group. So experienced genomicists and bioinformaticians working side by side, you can really get a key setting up a proper design of your sequencing experiment. You know, how many replicates, how much coverage do I need? Choosing the right products for the best outcome. You know, do I need a particular library prep method or DNA extraction method, things like that. You can offset the costs of the analysis and training to an experienced bioinformatician who can do it a lot quicker, which leaves the biological interpretation to the client, the user. And so the key here is saving time equals saving money. So I'm sure you've seen this before, but we've established the South Australian Genomic Centre last year, in July last year, and it's a statewide NCRIS supported genomics facility. So NCRIS is the National Research Infrastructure for Australia. It's a federal government money that flows through to research institutes and academic institutions. So it was established with a $7 million investment from all partners, $2 million of which came from Bioplatforms Australia, which is a subsidiary from NCRIS. So we're a fee-for-service genomics facility, and we have this integrated bioinformatics platform, and I'm also going to introduce my bioinformatics team in this as well. It's a partnership between SAMRI, the 3 universities of South Australia, the Australian Wine Research Institute, and also the National Australian Genome Research Facility, which we also work with. So we bring together expertise in not just human genomics or biomedical research, but also agriculture, so plant and livestock research, microbial and environmental genomics. It's a consolidation of a lot of existing facilities. The main node we have here at SAMRI, and smaller nodes we have at the University of Adelaide Waite Research Campus, which is more sort of the plant and agricultural sort of side, and also Flinders University. So this is the team here, the beautiful, lovely looking people. I just wanted to introduce, so David Lin, Professor David Lin is our scientific director. He's on the left-hand side of the left photo there. Just above him is Sen Wang, who's our facility manager. And really the people to the right here I'm going to focus on, which is our sort of bioinformatics team. And so we do lots of analysis services. We're looking to standardize analysis pipelines, collaborative and custom research projects, and also grant consultation. So really when you produce a data set, whether it be DNA sequencing, RNA, metagenomics, epigenomics, things like that, you produce a particular data set that needs a tool or a program to be run, or a pipeline or workflow that can really reduce the complexity of the incredibly complex data it is and produce a result. And these are just examples of some sort of projects that we've contributed to that have really looked at different aspects of this particular work. And so in our work, we use a sort of standardized community-based pipelines, many that are just available through the community, but also others that we've developed ourselves. So, you know, sequencing quality control, variant calling in various aspects, gene fusion structural rearrangement detection we've done in collaboration with Professor Deb White's group at SAMRI, RNA sequencing, single-cell RNA sequencing, all that sort of things as well. We generally rely on workflow language, including Nextflow and SnakeMate. So in terms of the team expertise, myself, I've got a sort of long research experience in reproductive genomics, epigenomics, and transcriptomics. I've worked across lots of different sort of plants, fungi, bacteria, and human research. Nathan is our sort of plant expert, and he is stationed at the Waite Research Campus a couple of days a week. And he's really got this amazing experience in genome informatics, genome assembly, and long read sequencing technology, a little bit like what Mark was talking about in the previous talk, and sort of plant genomics. Nee is one of our newest recruits, and she's got this amazing experience in transcriptomics. She runs all of our single-cell sequencing, single-cell RNA-seq projects, and she's doing an amazing job, and she's really our visualization guru. We do also have another position that's starting in April, May that we've just finished advertising for. So really, there's the product of running a pipeline, getting an answer, and we can give it to you straight away. But there's also opportunities to sort of collaborate and develop custom pipelines and things like that that are really important for sort of high impact research publications. And we work with a lot of different teams in this particular aspect. So just some areas in which our team has worked in previously, Nathan's done a lot of things in phylogenetics, and genome assembly projects, whether it be bacteria, plants, animals as such. I've done a lot of work in sort of comparative genomics and reproductive technology work. And we've just really started to do a whole lot of work in single cell sequencing with the 10x platform that we have at the SAGC, and Nee's really leading a lot of that work. So that's about it in terms of sort of highlighting all the different aspects of bioinformatics within the SAGC. I hope I've convinced you that sort of a sequencing and analysis service is really the way to go in terms of getting the best outcomes you can. Our website is sa-genomics.com.au, and we've also got sagc@samri.com as our email address. Feel free to email me or anyone within the team in terms of setting up a consultation. We can meet and go through different things. And yes, thank you very much. And thanks ABMC for having me.
Thanks, Jimmy. Great presentation. I really appreciate that. When is the best time to talk to a bioinformatician when it comes to designing your experiments? Beginning or the end?
At the beginning, preferably. I mean, as many people has shown before, you get really in bioinformatics and high throughput sequencing, any sort of complex assay, it's really garbage in and garbage out if you can control a lot of the factors that are involved in the sequencing aspect. As Mark would probably tell you, there's a lot of batch control, there's a lot of different biases that come in with any sort of high throughput technology. And really the expertise is knowing how to combat those. And so if you talk to a person in that particular respect, as soon as you can, we can sort of design ways of that. I mean, I've had lots of arguments with biologists about how many replicates to use in projects. So
it's one, isn't it? It's always only just one.
I also did ancient DNA work in which an N of 1 is optimal. But yeah, as soon as possible is the answer.
So Jimmy, I might have missed it, but also I'm wondering if you can mention what your training and activities might be also with the SAGC. I know you're running a few courses.
Yeah, so we've at the moment we've got 4 workshops designed for this year, and those are really focused on specific genomic technology. So our first one was with Professor Rob Edwards at Flinders in metagenomics, which we were really pleased that he wanted to partner with us to present that workshop. It went really well. Our next one we have at the Waite campus hosted by Australian Wine Research Institute, where it's going to be led by Nathan, and we're going to be talking about genome assembly and a lot of those particular aspects. Then we're going to have an RNA-seq and single-cell RNA-seq session, which is going to be at SAMRI mid-year, and then finishing off with epigenomics. We're looking at using this first year of this project to really gauge what sort of aspect that a lot of people require in their training, and the fact, as I mentioned, there's very few resources available within South Australia to train researchers in different aspects of bioinformatics, which are kind of crucial to what they do. So we're really looking to expand that, given the need, and we're going to be governed by the people, sort of their feedback and that. But yeah, it's a great question.
We've come to the hour, and I wanted just to thank both Mark and Jimmy for fantastic presentations, also for the team, Tamara and Yvette, and the team at SAMRI for organising the live feed that we have here. But I just wanted to remind you before that we close, is that these mini reviews are actually recorded and are available on the Adelaide Biomed City website. So if you go to the Minireview webpage, it's live on www.adelaidebiomed.com under the banner webinars, and where you can also watch this one and other past recordings as well. So I'd encourage you to do that, particularly if there was an aspect of the presentation today that you want to pick up on again. So that leaves me with one final task, in that thank you for joining us, and please join us next week at the same bat time and same bat channel. And for those old enough to remember, that is a reference to Batman. So cheerio.